For introduction, DNA profiling, also known as DNA fingerprinting, is a forensic technique used to identify individuals by analyzing their unique DNA sequence. It's worked by comparing certain regions of an individual DNA that are known to vary among individuals, called genetic markers. In forensic analysis, DNA profiling is often used to match DNA samples from a crime scene to those of suspect or victim. Freeman S. 2023. In summary, DNA profiling is a part of forensic analysis that makes investigation of crime more efficient. Next, for objective, first is to understand how biotechnology contribute to forensic DNA analysis, to understand the advantage and disadvantage of DNA analysis, and lastly, to understand DNA profiling in forensic DNA analysis.
The polymerase chain reaction is used in the forensic analysis. A PCR process involves several steps that are crucial for amplifying specific segments of DNA. The first step in PCR process is the denaturation. In this process, denaturation involves heating the double-stranded DNA template to separate it into two single strands. This process breaks the hydrogen bonds holding the DNA strands together for them to separate. Second step in PCR process is annealing. After denaturation, the temperature is lowered to enable the DNA primers to attach to the template DNA. DNA primers are short stretches of DNA or RNA that bind to specific regions on the DNA template. These steps allows the primers to mark the starting point of DNA replication. The last step in PCR process is the extension. In the extension step, the temperature is raised again and the DNA polymerase enzyme adds nucleotide bases, which is ATCG, to the DNA template and synthesizes a new complementary strand of DNA. The process results in the creation of a new double-stranded DNA molecule from the original single strands.
In the investigation of a crime scene where the murder of a victim has occurred, a forensic analysis involves the extraction of DNA samples from various sources, including fingerprints left at the scenes. Once the DNA is extracted from the suspect fingerprints, it undergoes polymerase chain reactions, a process used to amplify a specific regions of DNA for analysis. This amplified DNA from the suspect fingerprints is then compared to DNA samples obtained from the crime scenes. Upon the comparison, if the DNA extracted from the suspect fingerprints, which is referred to as suspect 2, matches the DNA sample found at the crime scenes, it is strongly suggests that involvement of suspect 2 in the crime. This match indicates significant connections between suspect 2 and the scene of the crime, raising the suspicion regarding the potential role in the murder of the victim. The matching DNA profiles provide a crucial evidence linking suspect 2 to the crime, potentially implicating them as the prime suspect in this investigation.
As for discussion, biotechnology used by forensic scientists to collect or process trace evidence such as hair, skin, blood, or semen samples found in crime scenes, specifically for now about DNA profiling that is obtained from fingerprint. As we know, PCR is crucial in DNA analysis as it amplifies tiny amounts of DNA from crime scenes, which helps to create DNA profile to identify suspects and also linking crimes. DNA profiling is a vital part in DNA analysis, which involves in analyzing specific parts of a person's DNA. In forensic analysis, profile is compared with DNA from crime scenes or suspects to help identify suspects or if there are any linkage to the crime scenes. As for the advantage of PCR in forensic DNA analysis, firstly, it is highly specific. This is because the PCR can distinguish DNA sequences just by one nucleotide, which will make it a very accurate technique. And the second one, it is very sensitive. PCR is a very useful technique when the amount of DNA sample is limited because it allows the detection of even a single copy of the specific DNA template. As for the disadvantage for PCR in forensic DNA analysis, first one is contamination. This is because the PCR technique is very susceptible to contamination from other sources of DNA or RNA or even the environment, which will mislead the data interpretation. And the second one is the errors in amplification. This is because the base substitution, indels, and also other alternation in DNA sequences can lead to inaccurate amplification and also which will make it false results.
In summary, the application of biotechnology in forensic investigation has proven to be an important tool for forensic experts as it allows for the analysis of biological evidence and solving major cases using DNA analysis with a high degree of accuracy and precision. Other than that, it can also enable the forensic expert to uncover critical information that may have otherwise remained hidden. That's all from us. Thank you.